So good morning, everyone. We are proceeding towards the 7th day of faculty development program entitled Exploring Multidisciplinary Avenues in Pharmaceutical Education and Research. Yesterday, we were having 3 lectures. The first lecture was on informatics-based development of animal models for translational pharmacological outcomes by Professor R.K. Goyal. The second and third lectures entitled Importance of Translational Research in Pharmaceutical Science by Professor Subhit Jain and IPRs and Patent Filing Procedures in India by Ms. Bhavna Mittal were very informative. So now we should proceed towards today's proceedings. So I would like to invite Dr. Divneet Kaur to deliver the welcome address.
Good morning, everyone. Today is the 7th day of faculty development program entitled Exploring Multidisciplinary Avenues in Pharmaceutical Education and Research. Today we have 3 technical sessions. For first session, I would like to welcome Dr. Harinder Singh, Principal Scientist, Bright Life Care Private Limited, Gurgaon. For second session, we have Dr. Kesar Raza from Department of Pharmacy, Central University of Rajasthan, Ajmer. I extend my warm welcome to you, sir. For third session, we have with us Mr. Amit Bhatia, Product Technical Specialist. I cherish the auspicious presence of Professor Dr. Indupal Kaur, Professor and Chairperson, UIPS, Punjab University, Chandigarh, who is our guest of honor for today. I also welcome Dr. V.K. Kapoor, Professor Adjunct, GHG Khalsa College of Pharmacy, former Dean and Chairperson, UIPS, Punjab University, Chandigarh. I would also like to welcome our worthy Principal, Professor Dr. Satvinder Kaur, GHG Khalsa College of Pharmacy. And I also welcome all the delegates and faculty members. So now we are going to start our session. I hand over the mic to Dr. Anuja for further proceedings.
Thank you very much, Dr. Divneet. So we have amongst us Professor Indupal Kaur, Professor and Chairperson, University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh. Professor Indupal Kaur was a former Director, Sophisticated Analytical Instrumentation Facility, Punjab University. She was also a former Dean, Faculty of Pharmaceutical Sciences, and presently she is also a Secretary, Indian Pharmaceutical Association, Punjab branch. She is amongst the top 2% most cited scientists across the globe as per Stanford University, USA. Her research forte is enhancing bioperformance of drugs, plant extracts, phytochemicals, and biomolecules, and probiotics using active tailored solid lipid nanoparticles, nanovesicles, and electrospun scaffolds. She has produced 26 PhDs and 55 postgraduates and has received funding of around 100 million Indian rupees from government and industry. She has delivered 50 invited talks and published around 150 articles with H-index of 45. She is a US Fulbright Fellow and was awarded Women Scientist Award by OPPI. She was graced with Break Technology Exposition Award consecutively for 2 years and Researcher of the Year Award by Sunpure Research Incubation Center. Recently, she won the Grand Innovation Challenge conducted by Indian Pharmaceutical Alliance in November 2021. Emphasis of her work lies on industrial and clinical translation and is reflected through licensing of 4 of her technologies to industry and 33 filed patent applications, out of which 11 are granted. So I invite Professor Indupal Kaur for the formal address. Please, ma'am.
Thank you so much for a very kind introduction. And it is, of course, a pleasure to be here. Respected Professor Kapoor is here, who's been our teacher and has rightly pointed out, he was the former chairperson and dean at UIPS. And I see so many of my students and alumnus today on this forum. And we have Dr. Satwinder, who's an alumnus. And I see two of my very dear students today, Dr. Parneet Kaur, who is a faculty with this college, and Dr. Harinder, who's going to be the next speaker. And also Kaisar Raza is also a very dear student. So of course, it is a pleasure. And when Parneet told me ki ma'am aap aao, so I couldn't say no to this opportunity. And the concept which has been put forth for this FDP, which is the multidisciplinarity of pharmaceutical sciences. I think we working in the area of pharmaceutical sciences have a very unique position where we have core branches, right from formulation technology to chemistry, medicinal chemistry, to preclinical evaluation and pharmacology, and the natural products, natural molecules, and their extraction, all put together. And if we can work in consonance, I think there could be answer to so many problems. And beyond that, now we have diversified into so many other disciplines in the area of pharmaceutical sciences, which all club together to basically, I would say, to identify a research problem, to find a solution to it, discover a molecule, then check for its performance in vivo in preclinical trials, and then try and develop a formulation which would be patient compliant and which can address any issues with which the pure drug molecule might have, which would lead to its abandonment rather than being taken up. And again, the importance of natural products in pharmacognosy cannot be undermined, in the sense that 60%, I think, or more of all the drug molecules which have been to date introduced have all come from the natural resources. The skeleton has been natural. It could be semi-synthetic, or it could be purely natural. And I think हम सब कुछ पढ़ते हैं बड़ी बारी. While I was younger, I would say pharmacy is a profession which is jack of all trades. But now I want to, I would say, commit and also claim that we are an all-in-one. So we have the basic knowledge of all those things. And though we start specializing and then super specializing with our PhD programs, but still, since we have that basic knowledge, and at the same time, there is a lot in pharmaceutical sciences which, gives us answers to much more things beyond what we are doing, even the natural phenomena and, the natural things which are there. Many a times when I discuss with my students, I say that we learn a lot. Achar mein namak zyada hota hai, which leads to osmosis, and that is how it protects it from getting, I would say, going bad over several years. Hamari dadi nanis usme khub sara tel dal dete the, and that oil, created anaerobic conditions, and the organisms would not grow. So the pharmacy has answers to several skill sets which are of use in day-to-day activities. I think the next lecture we have is on nutraceuticals, and right from the times of Hippocrates, it has been said that let food be thy medicine. And so we are talking of things where we can modify or improvise the food and the nutrition to manage diseases. So sky is the limit to do, to perform in pharmaceutical sciences, whether it is research, whether it is teaching, whether it is industry, whether it is hospital or community pharmacy, or whether it is drug regulatory, drug control. So there is a lot which we can do, and a lot with which we are equipped because of the multidisciplinarity of pharmaceutical sciences. So these are a few thoughts or ideas I had, and some of my messages to the young colleagues who are probably the participants is read, 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 and read. There is no alternative to reading in teaching or research. The more you learn, the more you dissipate, the more you can transfer, and the better you can do in the research. And then second thing is you have to be sincere towards science, towards the data you produce. The integrity in science is something which is very, very important, is key to any performance. And imagination, try and run it as wild as you can. Aspire for as big as you can, but at the same time, try and connect it with the reality, with the type of resources you have, the type of infrastructure you have, the type of capabilities and competency you have. And if you aspire for something where you lack, try and collaborate, try and connect with people, try and go out of your comfort zone, meet, connect, work together, and try and find answers. And as pharmaceutical scientists, and with the era of infections coming in, there is a lot which we can do. And in COVID times, everything failed, every profession failed, everybody felt the downstream of economy, except the profession of pharmaceutical sciences. So we are lucky, as well as we should be proud of our profession, and we should try and bring about change in our thought process. Knowledge, information, पढ़ाई का तभी सब मायने है जब हम सोचना उसके beyond शुरू कर देते हैं. Let not things, ideas, or information limit you. Let it help you to grow as human beings, as researchers, as scientists, as philosophers. Everywhere, I think we, by default, we fall in the category of top 5% across the world who are educated. So most of them are masters or maybe doctorates here. So maybe we are amongst the 1% of the global population who are as educated as we are. So we shoulder more responsibilities as well when we fall in this category of being well educated. So these are a few thoughts I had in mind, which come to my mind. I don't know if I could, deliver what was required of me, but this is what I thought I could share with all of you. Thank you. And hello, Harinder, and it is so nice to see you here today.
Yeah. Thank you, ma'am.
Ma'am, you are very, very much always required, because you add to the grace of our programs. And Dr. Bhardwaj says, sends his regards to you. He just called. I told him ki Dr.
morning,
Indu is
sir.
on the screen.
Right.
He said, pay my regards to her.
Sure.
Yeah.
Thank you.
Thank you. Thank you for being with us.
Thank you.
Thank you, ma'am. Thanks a lot for your kind words. So it's time to felicitate Professor Indupar Kaur. Ma'am, kindly accept our token of gratitude. Thank you, ma'am. Thanks a lot. So now we are having amongst us Dr. Harinder Singh as a resource person for first technical session. Dr. Harinder Singh has done his B.Pharm, M.Pharm and PhD from University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh. After PhD, he has total 8 years industrial experience in formulation development in pharmaceutical and the nutraceutical industry. He is well acquainted with all stages of pharmaceutical product development, especially for tablets, capsules, and pellets dosage forms. He is well versed with the development of nutraceutical and health supplements, including proteins, pre-workouts, and plant-based nutraceutical products. He has filed 12 patent applications, 9 Indian and 3 US patent applications. He has been granted 1 Indian patent and 1 US patent. He has been awarded with several awards in the scientific poster and oral presentations. Currently, he is working as principal scientist in formulation development at Bright Life Care Private Limited, R&D, Gurugram, Haryana. So I invite Professor Harinder Singh to kindly deliver his talk on nutraceuticals, a suitable choice for pharmaceuticals. Please.
Yeah, yeah. Thank you, Dr. Praneeth. So, good morning, everyone. I would like to thank to the college faculty and the management for giving me this opportunity. And I am thankful to my teachers, Kapoor sir and Indu ma'am, for guiding us. And I am highly thankful to Indu ma'am for guiding me in my PhD. It is the result of her hard work and untiring efforts and guidance what we are today. So it's because of all her efforts we are today. So coming to the topic. So is my screen visible?
It's visible.
Okay. So nutraceuticals, a suitable choice for pharmaceuticals. So as Induyam said that the famous scientist, father of medicine, Hippocrates said that let thy food be the medicine and medicine be thy food. Like we should have our food as a medicine. Otherwise, we have to eat our medicines as food. So various civilizations like Indian, Egyptian, Chinese, and Sumerian civilizations have already evidenced the use of food as medicine. In our India, as per the Ayurveda, the 5,000-year-old health science, which already have mentioned the use of various foods for various therapeutic uses. So first of all, what are nutraceuticals? So these are the non-toxic components which are extracted from food with a scientifically proven health benefit for the treatment or prevention of various diseases. So the term was coined by Stephen De Felici, who was the founder and MD of Foundation of Innovative Medicine. And the term comes from the combination of two words like nutrition and pharmaceutical, from where the term is derived, nutraceuticals. It covers all the ingredients, nutrients which are isolated from the plants or various foods, dietary supplements, fortified foods, functional foods, and medical foods. So nutraceuticals can be redefined as part of food or whole food, which have specific health benefit or medical benefit for treatment or prevention of diseases. So why the nutraceuticals are a suitable choice for pharmaceuticals? So first of all, we should understand the difference between the two. Whereas both are meant for treatment or prevention of diseases, but nutraceuticals have a low potency, and whereas pharmaceuticals have high potency. So the therapeutic action is immediate in case of pharmaceuticals, whereas in case of nutraceuticals, it takes a time. And in case of nutraceuticals, the interaction between the target receptors is weak. So the effect takes a long time to occur. Whereas in case of pharmaceuticals, it has a strong interaction with the target receptors. That's why they have the immediate effect. Whereas both have physiological mechanisms for their potential benefit. But in case of pharmaceuticals, the effect is more specific. Whereas in case of nutraceuticals, there is a multiple pathways of action which come into place and leading to a proper health benefit. Um, and in case of nutraceuticals, these are meant for inhibition of disease or prevention of disease, whereas the pharmaceuticals are meant for treatment of diseases. As I earlier said, that nutraceutical takes long-term application for its effects, whereas the pharmaceuticals have a short duration of action and they take short time for their effect. Whereas both are experimental and science-based approaches, but nutraceuticals have a low causal evidence, as the relationship between their activity and their effect are not well documented, whereas in case of pharmaceuticals, all the drug molecules which are approved, they have a strong cause and effect relationship. So nutraceuticals in India, there is an agency called FSCI, which regulate the nutraceutical market. For decades, the FDA had been regulating the nutrition supplements in India. But in 2006, the Indian government passed the act called as Food Safety and Standard Act, which streamlined all the regulations which were present in the market, comprising nutraceutical, foods, and dietary supplement regulations. And this act led to the formation of Food Safety and Standard Authority of India, which is known as FSCI. So the creation of FSCI, which this led to the registration and licensing of all food businesses and making them accountable for the products which they manufacture. It has all benefits like alignment of all international guidelines and regulations, so that our Indian products are as competitive as per the global products. And FSCI ensures the science-based standards for characterization of various nutraceutical products which are present in the market. And it has also brought clarity and uniformity on novel food areas, and also helping to curb the corruption. So as I earlier told, the nutraceuticals are various dietary supplements, micronutrients, functional foods, terpenoids, glucosinates, polyphenols. All these are derived from plants and natural sources and meant for various therapeutic uses. So coming to dietary supplements, so any product other than tobacco which is intended to supplement the diet and contain one or more of the following vitamins, minerals, herbal extracts, amino acids, or other nutrients which are derived from plants or natural sources. Coming to the fortified foods, these are the foods which are enriched with the vitamins and minerals. There is a limit for allowance of vitamin and minerals, which is called dietary reference intake or routine dietary allowance. So any food can have up to 100% of the value of the vitamin and minerals. No product can exceed the 100% value of recommended vitamin and mineral dosage on daily basis. So, and in case of various products, there is a loss of vitamins and minerals, especially in case of B vitamins. So there is a law that fortification should be up to a level so that it replaces the nutrients lost during the processes. Like vitamin B are lost in case of baked goods. So additional or extra vitamin quantity is added to compensate the loss. And various breakfast cereals has been fortified since 1940s to have various health benefits. Now coming to functional foods. So these are the foods which have a special health benefit beyond the traditional nutrients like vitamin and minerals. So these are the foods like fish, beans, whole grains, infant formulas. And now coming to the medical foods. Medical foods which are formulated for special medical need. Like in case of cancer patients, they have a specific diet requirement, like their diet should be high in protein so that they can tolerate the side effects of chemotherapy. Various diabetic diets, and in simple language like ORS. ORS is a medical food that is used to rehydrate the body in case of chronic diarrhea. So in the recent years, the medical cost has been significantly increased, which has led to people trying various options like fruits, vegetables, and consuming in excess to have the various nutrients in their diet. And from their diet, they are having these nutrients. But what kind of nutrients they are having from their diet is not clearly understood by them, because they don't have the proper knowledge about the nutrition. So nowadays there is a trend of hiring a nutritionist for their daily regular diet. Because nutraceuticals do possess multiple health benefits which are claimed by the various manufacturers of the products, and most nutraceuticals are currently known vital nutrients to the human body. But as I earlier said, we cannot have all the effects in one go, because there are various drug interactions, nutraceutical drug interaction, and their effects on different individuals having different health conditions is not described. So currently all the products which are available in the market, so they have not been checked for their bioavailability, because if the product is not bioavailable, then it will not produce the required health benefit. So the potential benefit of many nutraceuticals are not really optimized. So this may be due to relatively low or variable oral bioavailability, which may be due to the formation of insoluble complexes in the GIT. We all know that how the nutraceutical or a drug is behaving outside the body, but there are very much less studies on these nutraceuticals when they are consumed, and what is happening to the nutraceutical or the active molecule inside the body. It is not clearly understood, and very few studies are there in the scientific literature. So the low bioavailability reason may be due to the low solubility of the active in the gastrointestinal fluids, low permeability across the mucus layer or epithelial cells in the GIT. There might be any molecular transformation in the GIT which reduces the bioavailability of the nutrient, and also restricted liberation from the food matrix, like the active molecules from the food which is not liberated in the GIT cannot be absorbed in the system. So this reduced bioavailability, stability, permeability of the bioactive components in the GI fluids leads to their partial absorption, and also this may be due to the first pass metabolism, which is another reason for the low bioavailability. This results in their reduced or almost no biological activity, which is a major concern of the researchers today. Because if the compound or the active molecule is not properly absorbed in the body, then how it will show the therapeutic effect? And majority of the products which are present in the market are not standardized for their biological activity. So in the conventional mode of delivery of nutraceuticals, a small portion of the active compound is absorbed and reaches its biological site inside the body, and that is not significant enough to generate a therapeutic response. Rest of the nutraceutical compound is excreted through the body, and that may lead, and also it may lead to generate the non-specific toxicity or adverse side effects owing to undesirable biodistribution. As the targeting is not specific, then it may lead to generation of non-specific side effects. So we need to find a solution to resolve this problem so that we can have the nutraceutical benefit as per claimed by the manufacturer. So we can have the various delivery systems like effective solubilization, encapsulation in the delivery system, which leads to absorption in low doses, reduced frequency of administration, amended therapeutic index also. So there is a classification for the drug molecules, like biopharmaceutical classification scheme, which is widely used to characterize the various factors which are involved in the viability of drug molecules. But in case of nutraceuticals, this BCS classification is not clearly related to nutraceuticals, because there are various factors present, like the active molecule is entrapped inside a food matrix, so which limits the usefulness of the BCS classification for food components. So as you all know, this is a BCS classification system, whereas the drug molecule can have high solubility, high permeability, low solubility, high permeability. So likewise, nutraceuticals have also the same criteria, that they can be having high solubility, high permeability, and low solubility and high permeability. So scientists have devised a new method of classifying the nutraceuticals based on their characteristics, like bioaccessibility, in which the liberation from the food matrix, and after liberation from the food matrix is solubilization in the gastrointestinal fluids and its interaction with the various ions and molecules present in the GI fluids. Second comes the absorption. Absorption occurs at the mucous layers. So the active compound reacts with the mucous layer, how it reacts, and how the transport is mediated. It is mediated through the tight junction transport, or active transporters, or reflex transporters, and it should have a good permeability also. Transformation, these are of two types, like chemical degradation. When the active molecule is dissolved in the GI fluids, it may be chemically degraded to an inactive molecule, or and metabolism also. In the first metabolism, whereas most of the active molecules are converted to inactive molecules which have no therapeutic activity. So from this diagram, you can see that liberation from the food matrix is necessary for solubilization of active molecule in the GI fluids, and there onwards is absorption in the system. If the nutraceutical molecule makes an insoluble complex by interacting with the GI fluids, then it would not be absorbed, and there would be no therapeutic activity of the molecule. So the figure represents the various factors which limit the absorption of nutraceuticals, that is transport across the mucous layer. It depends upon the active molecule, that what kind of transport it undergoes. It is undergoing passive transport or active transport, or it is permeating through the tight junction. So the table summarizes all the limiting factors which affect the bioavailability. So the bioaccessibility in liberation limited, solubility limited, interaction limited. In case of absorption, mucin layer transport, tight junction transport, membrane permeability limited, active transport, efflux transport, transformation, chemical degradation, metabolism limited. So to resolve this various problems, again, the nutraceuticals rely on the pharmaceuticals. Like nanotechnology is basically derived from pharmaceutical delivery systems. So it's a delivery system in which the product active molecules are present in a nano form or entrapped in a nanocarrier system, and it would be helpful to increase the solubility, bioavailability, protecting against the various environmental hazards, enables sustained release, influence texture and flavor, targeted smart delivery. So the nutrients or the natural compound can be loaded into biocompatible, biodegradable nanoparticles. And as I earlier said, that it will improve the stability, solubility, bioavailability, and target specificity. So this is a pictorial representation of the nutraceutical converting into the nanoceuticals. So like these nanoparticles can be prepared by various methods, stirring, heating, ultrasonation, radiation. So these all are the various dosage form of nanoparticles. So the most important example of this nanotechnology is the incredible enhancement of solubility and bioavailability of curcumin. Curcumin, as we all know, is a wonder molecule which has been used since ages. We have been using curcumin in our household in raw form, but scientists have extracted the pure curcumin from the turmeric and incorporated it into the delivery system, which enhances the solubility and bioavailability of curcumin to several folds. And curcumin is the most researched molecules which are derived from the plant materials. Curcumin is having anti-cancer potential, which have been validated by the scientists in vitro and in vivo studies. And also if we incorporate the curcumin into magnetic nanoparticles, then we can have a site-specific effect of curcumin as in synergistic with the magnetic hyperthermia are demonstrated. There is another nutraceutical molecule, lutein, which is very beneficial for the eyesight and eye health. So it is incorporated into polymeric nanoparticles, which enhances this solubility, and its therapeutic action is more enhanced as compared to the simple lutein, which is not soluble. So there is a market formulation using nanolutein and curcumin also, which have been explored for cosmetic applications. So in conclusion, that nutraceuticals and functional foods, which have been developed with distinct trials, would be a potential to deliver unique products to the world at large. And development of better characterized research-proven molecules will help to enhance the consumer confidence in nutraceutical and functional foods products in the rest of the world. Also, nanotechnology can help in the effective delivery of nutraceuticals, and nutraceutical can be a better option for healthy life. In a nutshell, that nutraceuticals can be helpful in the modern days, like where the pollution and various contaminated food products which are causing the various health hazards. Among them, the cancer is top one. So a common people can rely on the nutraceuticals for living a quality life, improving the quality of life they are living. So nutraceuticals can be helpful for preventing diseases. Thank you so much.
Thank you, sir. Thanks a lot. It was really a very interesting and wonderful session. You have discussed state and scope of nutraceuticals in the current pharmaceutical market. You very well highlighted merits and demerits of nutraceuticals with relevant and suitable practical examples. Now the session is open for the queries. If anyone is having any kind of query, he or she can ask. We will unmute you. Anyone from the audience? I think, sir, there is no query from the audience as you have discussed very clearly all the things.
So I would like to mention one more thing, that students can also pursue a career in nutraceuticals or nutrition industry, and pharmacy students are more apt for the food industry because there are no regulation and standards in the food industry. So pharmaceutical students are more educated to follow the guidelines and making the products as per GMP standard. So you can guide the students to pursue the career in nutraceuticals or nutrition industry also.
Okay, sir.
Hello, hello.
Yes, sir. Kapoor, sir. Hanji.
Am I audible? Am I audible?
Yes, sir.
Yes,
You are
sir.
audible.
Very nice, Sarvinder. Though I have never met you, but it is nice to interact with you by your very enlightening lecture on nutraceuticals, right? And it was very informative. And nutraceuticals is an emerging area where the pharmacy research can be carried out more effectively. See, there are a number of metabolic disorder diseases for which there is no treatment in allopathic system. The only treatment available for metabolic disorder diseases is a specialized food which is imported and not made in India to relieve the system. I think there is a very good scope of doing research in the area of metabolic disorder diseases and go into the composition of the imported food. What are the constituents, what are the items present in that food, which allege of the disease toxicity. So I don't think anybody else is delving in this area of research, but there is a lot of scope why that food is imported, why that food cannot be manufactured in India, what is so special about that food for the metabolic disorder diseases. This is one thing. Secondly, since you are working in a company which is dealing with nutraceuticals, I have a query. Hello.
Yes, sir. I'm listening.
You see, soya beans is very extensively used as a health diet. So many soya products are, being prescribed by the doctors, or they are taken as such by women. And it has been found that soya contains some isoflavonoids like digoxin and genisteins, which resemble the molecule of estradiol. And they are also rightly called as phytoestrogens.
Yes, sir.
Now there is a controversy that the cancer of breast in women is mediated through a receptor on which estradiol and these isoflavonoids can also act. One of the theory is that these isoflavonoids or phytoestrogens will block the receptors because they resemble the structure of estradiol and thus prevent the onset of cancer. This is one theory. That is, they have been recommended for the prevention of breast cancer in the woman. But another theory is there, since these isoflavonoids, they resemble in structure with estradiol. By interacting with the receptor, they can initiate the process of cancer formation. So there are two extremes, which I think it has not been definitely resolved. Whether the soya and soya products can protect the woman from breast cancer, or it itself can cause breast cancer. This is my query. And if you have come across any literature of this, I shall be very happy to be enlightened on it. But your lecture was very informative, very interesting, and I enjoyed it. Congratulations.
Yeah, thank you, sir. Thank you so much. I have been part of a class you taught at UIPS. So you have taught us medicinal chemistry in the class.
Oh, I left actually in 2005.
Yes, sir. I was admitted in BPharm first year 2002. So you were our teacher there.
Okay. Then I must have taught you medicinal chemistry.
Yes, sir. Yes, sir.
Thank you very much for remembering me.
Yes, sir. We always remember, sir.
Thank you.
Sir, one my take on soya products is that soya has been in use in Indian food since ages. So and it's a very good source of protein. But it's only marketing which is funded by the various, I would say, pharmaceutical industries, like which are defaming the soya products. So the soya has been always a beneficial food for all, be it women or men. Today's lifestyle, pollution, contaminated food, and various other factors that are contributing to various metabolic diseases and the cancer also. So the industry is blaming the way what they can find easily target.
Sir, one more question is there. How to evaluate nutraceutical in the chemical sense by Ma'am Charulata?
Yeah, we can we analyze the drug by UV. So in the same case, all the nutraceuticals have active compounds which can be characterized using various instruments like UV, NMR, and HPLC, GC, depending upon the sensitivity of the equipment. Majorly, the molecules are characterized by UV and gravimetry.
Thank you, sir. Thank you, sir. Now it's time to facilitate Dr. Harinder Singh. Sir, kindly accept our token of gratitude.
Yeah, thank you so much.
Thank you, sir. Thank you again for being with us and sharing your knowledge with us. Thank you, sir.
Yeah, thank you so much. Thank you.
The speaker and molecular weight measure. He has a total work experience of 22 years in the field of analytical instruments and has been associated with Melbourne Panalytical since 2004. He has worked as a service engineer in the beginning of his career and then shifted to sales, and now currently he is in the product specialist role. He is a bachelor in engineering instrumentation. I invite Mr. Amit Bhatia for his talk. Please, sir.
Good afternoon, ma'am.
Good afternoon, sir.
Good afternoon, everyone. As ma'am introduced me, I am Amit Bhatia. So I'll just share my screen. And today we'll be discussing about dynamic light scattering. So I'll just share the screen with you. Is my screen visible, ma'am?
Yes, sir, it is visible.
Okay. So good afternoon, everyone. My name is Amit Bhatia, and as ma'am told, I am the product technical specialist for dynamic light scattering and gel permeation chromatography. So today what we will be doing is we'll be focusing on dynamic light scattering, its basics and the advancements that have happened in the years since the dynamic light scattering started as an instrumentation technique. So let us go into dynamic light scattering, how the dynamic light scattering technique has evolved over the years and how it has moved for the measurement of particle size and zeta potential. So the heart of any instrument is in a dynamic light scattering equipment is basically a correlator. So the first correlator that was invented was in the year 1971. And it took around 12 years for getting the first dynamic light scattering based instrument to be developed, which was developed by our company called Malvern Analytical Instruments. That was the first year in 1983 when we designed the Zeta Sizer, which was basically the dynamic light scattering instrument, which was used for the measurement of particle size and zeta potential. So over the years from 1983 till today, 2022, we have been making several advancements in the field of dynamic light scattering with the help of various technologies that have come up with time, which included some of them were like mixed mode measurement, which we would be discussing during the presentation, and how dynamic light scattering is useful for measuring particle size. Right? So the current day equipments look somewhat like this. So these are very much similar if you have worked in the laboratories with a UV visible spectrophotometer. So these look very much similar to spectrophotometers. You just click on this green part. This particular cuvette box will open. You can put your cuvettes here and you can do particle size measurement, zeta potential measurement, as well as molecular weight measurements. So all these measurements can be done with the help of dynamic light scattering based instruments. So what happens in a dynamic light scattering based instrument is just like you have a cuvette in your UV visible spectrophotometer, you basically have a cuvette in dynamic light scattering also. You allow your particles to be moving in Brownian motion in your cuvette. From the other side, you shine a laser. Now, since these particles are moving in Brownian motion, you are shining a laser from one side, and on 90 degrees or at one particular angle, you are measuring the fluctuation in the intensity of light that is happening because of the movement of the particles in the cuvette, right? So the intensity fluctuations when we measure with respect to time, that is called as dynamic light scattering. And when we average this over a period of time, that is called as static light scattering. So what we need to understand is that dynamic light scattering is used for the measurement of particle size, whereas static light scattering is used for the measurement of molecular weight measurements. So we basically measure in the instrument the intensity fluctuations of light with respect to time because of the Brownian movement of the particles in the cuvette. So this is how the instrument measures particle size. So what basically happens is, as I said, particles move due to Brownian motion. We measure fluctuations in the intensity of the scattered light with the help of a detector. From there, we calculate the translational diffusion coefficient. We put the value of translational diffusion coefficient, which is nothing but D, in the Stokes-Einstein equation. From there, we calculate the hydrodynamic diameter, and by applying various algorithms, we are able to get the size distribution. So if everyone is interested in nanoparticle size analysis, this is how the measurement is done. You keep your molecules in your, you suspend them in a particular cuvette, in a certain solvent in a cuvette. You allow the particles to move in Brownian motion. On the detector, we measure the intensity of the fluctuation of the light. From there, we calculate the translational diffusion coefficient D, which we put into the Stokes-Einstein equation and calculate the Z average, which is nothing but the nanoparticle size. And by applying various algorithms, we get the size distribution for your particles. So when you are working on dynamic light scattering equipments, there are certain things which you have to take care. So when you put any sample into your machine, you need to know what is the upper size limit of your dynamic light scattering equipment. That is, what is the maximum size range that you can measure. So generally, when we say we are working on nanoparticles, we generally mean that we are working on particles which are in the range of say 100 to 500 nanometers. That is the range which we consider as the nanoparticle size range. So we need to know what is the upper size limit of the technology that we are using and what it is dependent upon. What is the lower size limit? That means what is the minimum size that you can measure. So when we say minimum size, what that means is we can go down up to say 0.3 nanometers, right? So with the instruments that are available, which are based on dynamic light scattering, you can measure up to 0.3 nanometers on the lower side. Similarly, what is the lower limit of concentration? That is whether I can go up to 0.1 mg per ml or not using the instrument. That is the lower limit of concentration. So what is dependent upon, we'll just check that. And what is the upper concentration range of the DLS technology? So these are the factors that we need to consider when we are doing measurements on a dynamic light scattering equipment, right? Now, assume you are working in a laboratory wherein you are in a central facility. So you don't know what is the concentration range of the samples that you would be receiving would be. So what you would be aiming at is that your instrument should be able to measure concentration ranges from minimum to maximum. So the widest concentration range that you can measure would be the most beneficial. So if you are say working on some protein and peptides where the concentrations are very low, to say where you're working on inkjet printers where the concentrations are up to 40% weight by volume. So the instrument should be able to measure from very low concentrations to very high concentrations. The instrument should be able to measure from lower size limits, say 0.3 nanometers, to up to higher size limits, that is say 10 microns, 11 microns, 6 microns. So even if those particles are there, those should be measured. So these are the parameters that we need to consider while we are working on particle sizing equipment. Now, what I will discuss in my next few slides is that what is the upper size limit of your technology dependent on, what is the lower size limit dependent on, similarly, what is the concentration lower limit dependent on, and what is the upper concentration limit of the technology dependent on. So as I said, the upper size limit is defined when the particle motion is not random. So assume you have large particles, say approximately 3 to 4 microns. Now what will happen in those cases is instead of the particles moving in Brownian motion in the cuvette, they will have a unidirectional motion. Why? Because the sedimentation is taking place. So for larger particles with higher density, the particles will start to move in a top to bottom approach, right? They will start moving from top of the cuvette to the bottom of the cuvette, which means that it is experiencing something which is sedimentation kind of a process. So whenever such kind of sedimentation process takes place in our particles, that defines the upper size limit of the machine, right? So if we have particles which are less dense materials, we can measure up to 15 microns. But if your particles are say like tungsten carbide, which is one of the densest materials, so in those cases you will not be able to measure even 200 nanometers with your machine. So even though all the manufacturers would be claiming certain ranges of particle size range, say from 0.3 nanometers to 10 microns. If you look at any of the specification sheets that are available for nanoparticle sizes, it would say 0.3 nanometers or say 1 nanometer to up to 10 microns. But what you have to understand is the upper limit of the machine depends upon your particular particle. It doesn't depend upon the range of the machine. If your particle is sedimenting, that defines the upper size limit of your machine, of your instrument. So higher density materials, the higher the density of the material, lower will be the particle size that you can measure. Lower the density of the material, higher will be the particle size range that you can cover. Right? So in terms of when the instrumentation companies define, we can measure up to 15 micron samples using DLS instruments. But again, these are under very specified conditions and with very specific sample types where the density is very low. Right? Similarly, the lower size limit of the instrument. So when I say that the lower size limit can go down up to 0.3 nanometers or say 1 nanometer, what it depends upon is, it depends upon the amount of excess scattered light that the sample generates as compared to the suspending medium. So suppose you have suspended your sample in water. So water in itself would be having certain amount of scattering that it produces. So the minimum amount of sample that you need to add so that your detector is able to detect the variation because of the addition of the particles into the solvent, that defines the lower size limit of the instrument. So when we say that we are able to measure up to 0.6 nanometers or 0.3 nanometers, what we do is we measure sucrose sample on the DLS instruments to show that our instrument can go down up to 0.3 nanometers. Right? So this is how the upper size limit and the lower size limit of a dynamic light scattering instrument is defined. So whenever you are looking at your particles with that you want to measure, you need to know what is the approximate density, because that will define the upper size limit, and what is the scattering. Now, when we say scattering, so assume you are having a gold nanoparticle. So the gold nanoparticle would be producing significant amount of scattering at say 20 nanometers as compared to any lysozyme sample, which produces lesser scattering at same particle size, right? So we need to know our particles to define what is the lower size limit and what is the upper size limit of our machine. Now, as I said, the other parameter that we need to see is the lower limit of concentration. So what is the lowest limit of concentration that we can measure? So as I said, we can measure with our instruments up to 0.1 mg per ml lysozyme sample on a DLS instrument. That is the minimum concentration that we can measure. What is it dependent on? It is dependent again on the amount of excess scattered light that the sample generates as compared to the suspending medium. So once you suspend your particles in a particular medium, it should develop certain amount of scattering that is produced so that it can be detected by the detector. Right? So this is what defines the lower limit of the concentration, and to have a physical value, 0.1 mg per ml is what is the minimum value of concentration that can be measured on the dynamic light scattering equipments. Now, similarly, there are certain upper limits of concentrations that are available. So if you look at the specification sheets, it would say 40% weight by volume. Now, the upper limit of the concentration, however, is dependent upon the multiple scattering effects and the particle-particle interactions. Now, these two should not be taking place in your cuvette. If these two are not taking place, then the upper limit of the concentration would be approximately 40% weight by volume. Now, anyone who is working with a dynamic light scattering equipment should understand what is multiple scattering, because when you understand multiple scattering, that will help you in defining that what is the concentration range on which you can work, right? So as I said, when you are working in a lab, say you're working in a central facility and there is a sample that comes to you, you are not sure what will be the kind of sample that you're receiving. There are certain things that you have to take care. One, what is the upper limit? What is the lower limit? What all are dependent upon that? I mean, what is the density of the material that is being measured? Similarly, what is the concentration range that is there? What is the minimum? What is the highest? So in order to understand the upper concentration limit of your machine, you need to understand what is multiple scattering. So when you see multiple scattering, as I said, this rectangular box that you see is basically representing the cuvette that is there in your machine. You, as I said, you shine laser from one side and you are measuring it at the detector, right? Now, if you have small particles in low concentration, what happens is once the laser hits a particle, the intensity fluctuation directly reaches onto the detector. So this is the condition that we require. This is the ideal condition. Now assume you have small particles at high concentration. So what will happen is once you shine the laser onto the cuvette, it hits a particular particle, and instead of it directly going onto the detector, it goes to hit another particle, then another particle, then another particle, and then it finally reaches onto the detector. So when such a thing happens, that is the place where we say multiple scattering is taking place. So this multiple hitting of the intensity fluctuation onto the various particles and then reaching onto the detector is called multiple scattering. So similar size particle, when they were in lower concentrations, you did not have a problem. You were measuring it very accurately. But as soon as small particles with higher concentrations, again, you had a problem because multiple scattering starts taking place. So what happens at multiple scattering is, in case of multiple scattering, your size would start reducing. Actually, your size would be say 80 nanometers, but because of the multiple scattering effects that are taking place, you would see the size as 60 nanometers, right? Which will not be accurate. So that is why you have to avoid multiple scattering. And this is how you define what is the low concentration limit to the higher concentration limit. The condition of multiple scattering should not take place. That is the main requirement. If you are working in a range of concentration where multiple scattering is not happening, then you will get more accurate results. So this was an example of small particles. Now assume you have large particles at low concentration. So again, there is no problem because the intensity fluctuation is directly reaching onto the detector. But if you have larger particles at higher concentration, again, multiple scattering takes place and then it reaches onto the detector, which would again give you the wrong results, right? So what we are seeing here is that the concentration range that we can work on depends on the onset of multiple scattering, right? So as you increase the concentration of your sample, if multiple scattering starts taking place at then you cannot do the measurements at those concentrations, right? So in the previous generation equipments, which were based on a 90-degree setup, when we say 90-degree, what does that mean? Your laser was shining from here and you were measuring the intensity fluctuation at 90 degrees, right? So with the original dynamic light scattering equipments, which initially used to come in the years, say 1990s, all of the setups were 90 degrees. So they were limited by the fact that you could not measure a wide concentration range of samples on those instruments, right? So how do we increase the concentration range? And why is it important to increase the concentration range? Because you have a single instrument in your lab. Now, if you're working on protein peptides kind of a sample, you would be getting very low concentrations. So the instrument should be working fine on low concentration also. Now, assume you are working in a, as I said, in a central facility in a university, and you get some high concentration samples. So say around 5% weight by volume or 20% weight by volume or up to 40% weight by volume. So at higher concentrations also your instrument should be working fine, right? And it should be giving you accurate results. So this is how the technology evolved from a 90-degree setup, and we have to move to something on which you could measure wider range of concentrations, right? So what we understood was multiple scattering effects must be minimized in order to work at higher concentrations. Now, in order to work at higher concentrations, one of the approaches is to minimize the path length over which the scattered light has to travel, which can be obtained by the backscatter optics. So what we do here is, instead of placing the detector at 90 degrees, the previous So in the previous slides, you could see you had a laser shining in this direction and you were detecting light at 90 degrees. So as the technology advanced and the applications that were being that we wanted to address using the dynamic light scattering instruments increased, we needed to increase our concentration range, right? So instead of placing the detector at 90 degrees, now we place the detector at 173 degrees. So this angle that you see is 173 degrees and this angle is 7 degrees, right? So instead of placing the detector at 90 degrees, we are placing the detector at 173 degrees. Now, what was the advantage of this? So if you had small particles or dilute samples, you could shine the laser, let the laser go into the cell center so that you are having sufficient amount of particles to be measured and allow the light to come back onto the detector from the cell center, right? Now, as we said in the previous slide, small particles at higher concentrations. So what used to happen is this light used to go to the cell center and we used to measure it at 90 degrees. Because we were measuring it at 90 degrees, there was multiple scattering that was happening. And then we were measuring it at 90 degrees. Now, with having a detector at 173 degrees, and by having a variable movement position, depending upon the concentration of the sample that you have put in, we take the measurement either from the cell center or from the cell wall, right? Now, if you have a higher concentration, you will be measuring it from the cell wall so that multiple scattering doesn't take place. If you are having dilute samples, you will be measuring it from the cell center so that you are able to measure sufficient number of particles which produce sufficient amount of intensity for the detector to detect the samples. So what we have done is, instead of having a fixed optics in which the laser was placed in this direction and the detector was at 90 degrees, we have now changed the detector position from 90 degrees to 173 degrees. And we have this focusing lens which keeps moving front and back depending on what kind of sample you are measuring. So if you have a concentrated sample in your machine, it would measure from the cell wall. If you have a lower concentration sample or a dilute sample, it would measure from the cell center. So what effectively it does is, in a 90-degree system, the usable concentration range was very small. And after this usable concentration range, which used to be 1% weight by volume, there was a decrease in the particle size that we would see because of the multiple scattering effects taking place. Now, with the non-invasive backscatter optics, we have increased the usable concentration range from 1% weight by volume to up to 40% weight by volume. And then the scattering pattern starts to decrease and it leads to multiple scattering effects. So what we are saying is, by having a non-invasive backscatter optics, that means by placing the detector at 173 degrees and having a movable focusing lens, we have been able to increase the range of concentrations that we could measure on the sample, right? So now with the same instrument, you can measure very low or very dilute samples to very highly concentrated samples. So this was the advancement in the technology that happened in the years early 2000s with the help of backscatter optics, which helped us in increasing the concentration range of the instrument. So with a single instrument, we could measure proteins, peptides, etc., which were at very low concentrations, to something like inkjet printers, wherein in a very small box, like in the printer, in a very small cartridge, you have very high concentrations of nanoparticles dispersed in it. So you could measure up to 40% weight by volume on the higher concentrations, and you could measure up to, say, 0.1 mg per ml on the lower sides when you are working with dilute samples or when you were working with protein kinds of samples, right? So all this was happening because of the non-invasive backscatter optics, and it helped us in measuring particles which are even lesser than 1000 Daltons, right? So the non-invasive backscatter optics, which included the 173-degree measurement angle, helps us in increasing the sensitivity, helps in increasing the low to high concentration range that you can measure, and it helps us in giving consistent results. That means you were able to get more repeatable results with the help of the non-invasive backscatter optics. So what we basically did was, we were moving the focusing lens. In case of dilute samples, we were measuring it at cell center. And in case of concentrated samples, we are measuring it close to the cell wall. right? So this movable optics position at 173-degree angle is nothing but what we say as non-invasive backscatter optics, which helps you in measuring wider range of concentrations, right? And the other advantage that we had was we were able to measure more sample volume by using the backscatter optics, which helped us in increasing the sensitivity of our instrument to up to 8 times more as compared to the 90-degree setup. So what we are basically trying to say here is that the original dynamic light scattering equipment instruments were all based on a 90-degree setup. But as the applications on which the nanoparticles were being used, as they diversified, there was a requirement to have wider range of concentrations to be measured on the same instrument, which was not possible using a 90-degree setup. So in order to overcome those, the advancements that took place in the technology helped us in developing a non-invasive backscatter optics at 173 degrees, which helped us in increasing the sensitivity by measuring more volume as compared to the 90-degree setup. And by measuring it at backscatter optics, we were able to increase the concentration range of our machine. So dilute samples were being measured at the center of the cuvette, and concentrated samples were being measured at the wall of the cuvette, right? So these were the various measurement positions that you could measure based on the concentration. Now this automatically is selected by the instrument. You don't have to select at what concentration, which measurement position has to be selected. You just have to put the cuvette in the dynamic light scattering based instrument, just like you put it in a UV visible spectrophotometer. All these optimizations, that at what concentration, which particular measurement position will be selected, is done by the software automatically and is taken care by the machine automatically. The main advantage of having this non-invasive backscatter optics is you are able to increase the concentration range, you are able to increase the sensitivity of the instrument, and you are able to get more repeatable measurements by using non-invasive backscatter optics, right? So this is how the optics move. So what happens is, depending on the concentration, your focusing lens moves automatically so that you are able to measure wide concentration ranges. So if you look at this setup, you have concentration on the x-axis and the average Z average on the y-axis. So if you look at the blue line, so from very low concentration ranges, which is 0.0001% weight by volume, you can go up to 40% weight by volume by using a blue line, that is the non-invasive backscatter optics. But if you look at the 90-degree setup, the red dots, if you see, even at 0.1% weight by volume, it started to fall. The Z average started to fall. And why it started to fall? Because your multiple scattering effects started to take place, and hence your 90-degree setup was not suitable. So that is how we moved from a 90-degree setup to a backscatter optics arrangement, so that we were able to measure a wider concentration range of samples. So I'll just like to take a break here and ask whether you have any questions about particle size right now.
Thank you, sir.
Because next we will be moving on to zeta potential. So when we go on to zeta potential measurements, that will be a separate topic. So any doubts that anyone has with respect to particle size measurement, how it is done, how what all parameters need to be checked, whether your sample will be suitable for DLS instrument or not? Any questions?
Dear participants, if you are having any problem or any query regarding the first section in the particle size determination, you can ask from sir. Anyone? Kindly post their query in the chat box. We will unmute you. Either you can raise your hand. I think, sir, everything is clear. You can move on the second section.
Okay, so we'll move on to the second section, which is basically zeta potential. So I'll just like to brief again what we had in the particle size. So as I said, a dynamic light scattering equipment is basically used for measurement of particle size, zeta potential, and to a certain extent molecular weight. So when we were doing particle size measurement, we need to remember that the dynamic light scattering equipment is usually used only for measurement of nanoparticles. What is the range of nanoparticles that we can measure depends totally on the kind of sample that you are giving to the instrument. So if you have a particle which is highly dense, the particle size range of the instrument is reduced to certain microns, say 1 micron or 2 microns. But if you are giving a material which is very less dense, you can measure up to 15 microns. Similarly, depending upon what kind of instrument you are using, so if you have a 90-degree setup, you can measure a concentration range of say up to 1% weight by volume. But if you are using a backscattering optics arrangement, you can measure from 0.0001% weight by volume, and you can go up to 40% by volume to address various kinds of applications that you may have in a central laboratory. Right now, the main consideration that we had in case of a dynamic light scattering instrument for doing particle size measurement was the onset of multiple scattering. So the onset of multiple scattering is very important, because once the multiple scattering starts to happen, your particle size tends to decrease because of the instrument effects, not because your actual particle size has reduced. So we have to take care that the multiple scattering doesn't take place in our machine whenever we are using it. So in order to increase the concentration range, we developed certain technologies which were called as non-invasive backscatter optics, which had a angle instead of 90 degrees, we were measuring it at 173 degrees, and we had a movable optics arrangement so that our concentration range of the instrument can be increased, and the same instrument could be used for wide range of applications. So this is what we have covered till now. And now we move on to the second part, which is basically the zeta potential. Now, as you are aware, zeta potential is something which helps us in determining the stability of the suspensions or the dispersions, right? So zeta potential is nothing but a parameter that is used for predicting the dispersion stability. Now, if you have a larger value of zeta potential, that means you have a more stable dispersion. So that means if you have a zeta potential value which is greater than plus 40 or plus 30, it will mean that that particular dispersion is stable. And if it has a value of less than minus 30, that means minus 40, minus 50, again, your dispersion will be stable. If your zeta potential value is close to the isoelectric point, which is basically 0 millivolts, in those cases, the zeta potential will be, say, if it is in between minus 30 to plus 30, that means your dispersion will not remain stable. So let us just look at the theory part of zeta potential and how the instruments measures the zeta potential. So dispersion stability, the stability of colloidal dispersions is determined by the sum of attractive forces, which is nothing but the van der Waals forces, and repulsive forces, which particles experience as they approach one another. Now, the stability of a particle dispersion can be lost through various mechanisms. So what we mean by that is, so if you have a stable dispersion, it may flocculate, coagulate, and sediment, and then phase separate. Or it may take a different route. It may sediment. After sedimentation, it may flocculate and coagulate, and then phase separate. Right? So what we are aiming at is that our stable system, a stable dispersion, should remain stable over a longer period of time. Right? It should not happen that your stable system phase disperses and becomes unstable. So in order to maintain our stable system, there is a parameter which is called as zeta potential that we have to monitor. So if the particles have sufficiently high repulsion, then the dispersion will resist flocculation. That means your dispersion will have higher stability. If there is no repulsive forces, then the flocculation or coagulation will eventually happen, and it would lead to dispersion instability. So how do we maintain dispersion stability? For dispersion stability, the repulsive forces must be dominant. So if you are having dominant repulsive forces in your dispersion, you will be able to maintain it in a stable condition. So the parameter that is used to check whether our dispersion will remain stable or not is nothing but the zeta potential. So what is zeta potential? the zeta potential is the overall charge that a particle acquires in a particular medium. So assume this is a negatively charged particle. So just outside this negatively charged particle, there will be a layer of positive charges, which will be there. Now, beyond this charge, there will be a slipping plane in which you will have particles which would be coming in and moving out, right? So you will be having positive and negative particles which will be moving in and coming out across this slipping plane. So zeta potential is nothing but the charge of the overall particle in a particular medium at the slipping plane, right? So zeta potential is the overall charge that a particle acquires in a particular medium. It is the potential at the slipping plane, not at the surface, but at the slipping plane. This we need to remember. So basically what we are saying is it is not dependent on the surface charge, but it is dependent on the surface charge and the medium in which it is dispersed in, right? So it is a combination of the surface charge as well as the medium in which it is dispersed in. So it depends both on the nature of the surface and the dispersant. It may be unrelated to the surface potential, and small changes in the value of pH or concentration of ions can lead to dramatic changes in zeta potential. Now, why this is important is because if you have prepared a dispersion, you need to look at the dispersion stability. Now, how you can control the value of zeta potential? You can control the values of zeta potential by varying the pH and by varying the concentration of the ions. So you have to optimize the pH and the concentrations of the ions in such a way that you get higher values of zeta potential, that is more than plus 30 or less than minus 30. So dispersion stability and zeta potential. If all the particles have large negative or positive zeta potential, they will repel each other and there will be dispersion stability. If the particles have low zeta potential values, then there is no force to prevent the particles from coming together. Eventually, it will lead to flocculation or coagulation, and that leads to dispersion instability, right? So what we are basically saying is, as a thumb rule, the dividing line is plus 30 and minus 30. So any value of zeta potential which is greater than plus 30, that means 35, 40, 45, 50, or less than minus 30, means minus 35, minus 40, minus 45. So anything which is further away from the isoelectric point means your dispersion stability will be more as compared to something which has a value of lesser zeta potential. So if you have two dispersions and you have to optimize which dispersion to be used, when you measure the zeta potential, if one of the dispersions gives you a value, say minus 20, the other gives you minus 40, you have to select the minus 41 because that will remain stable over a longer period of time, right? That is how you predict dispersion stability using zeta potential. So what all is zeta potential dependent on? The zeta potential, as I said, is dependent on the pH. It is dependent on the conductivity of the medium and the concentration of a particular additive in the product formulation. It could be an ionic surfactant or a concentration of any polymer. So as you vary these, your value of zeta potential varies. And what you have to basically do when you are formulating a particular dispersion is that you have to vary these parameters in such a way that you get a value of zeta potential which is as far as away from the isoelectric point. So basically, more away from the zero value, the more stable will be your dispersion. So you have to aim at making values of zeta potential higher than plus 30 and below minus 30 so that you make sure that your dispersion will remain stable. So this was the basic theory of zeta potential. Now, how the instrument measures zeta potential. So just like in case of a particle size, the main difficulty that we faced was multiple scattering. Now, because of multiple scattering, what used to happen? The size used to decrease, right? Because the intensity fluctuation was not reaching directly onto the detector, and hence its particle size was decreasing, even though the size was actually, say, 60 nanometers, but you could see only 40 nanometers because multiple scattering was starting to take place. Now, what considerations you have to do when you are measuring zeta potential? We'll just see that. What basically we have to do is we have to avoid electroosmosis from taking place in the measurement. If we are able to avoid electroosmosis effects, we are able to measure zeta potential more accurately. So what I'll do is I'll show you how zeta potential is measured in the instrument and what are the considerations that we need to take care. Right. So what happens in case of zeta potential is you have a cuvette which has two electrodes on the either side. Right. You apply electrical voltages onto those two electrodes and you force the particles in the cuvette to move in a particular direction. So what will happen is, assume you apply positive voltage. So the negatively charged particles will get attracted to it and the positively charged particles will move away from it. Right. So when you apply a certain amount of voltage, there will be certain movements that will happen in the movement of the particles. Those movements that are happening because of the application of an electric field will help you in measuring the velocity of the particles which move inside the cuvette. Once you are able to measure the velocity, you are able to measure the electrokinetic effects, which is basically the electrophoresis that we measure. And from there, you are able to measure the zeta potential. So when we see a zeta potential measurement, we basically apply voltages. So what we have to remember is an important consequence of the existence of electrical charges on surfaces is that we exhibit certain effects under the influence of applied electric field. So basically, when you have particles dispersed in a particular medium and you are applying a certain amount of voltage to it, what we are saying is they show certain electrokinetic effects. Now, these electrokinetic effects are mainly divided into electrophoresis and electroosmosis. What we have to remember is electrophoresis is the desired effect. Electroosmosis is the undesirable effect. So whenever we are doing zeta potential measurements, electrophoretic mobility is what we measure. Electroosmotic mobility is what we have to compensate, right? Because this is the desirable result and this is not the desirable result, right? So most of the equipments that are being designed are based on how to remove electroosmosis effects that happen when you apply certain voltages onto the particles that are dispersed in a particular medium. So in order to understand how electroosmosis is avoided, let us first understand what is electrophoresis and what is electroosmosis. So electrophoresis is nothing but the movement of the charged particles relative to the liquid that it is suspended in under the influence of an electric field. So we have to always remember anything that is happening for zeta potential measurement requires a certain amount of voltage to be applied, a certain amount of electric field to be applied. So what is electrophoresis? It is the movement of charged particles relative to the liquid that it is suspended in. And what is electroosmosis? It is the movement of the liquid itself relative to the stationary charged surface. Now, where does the stationary charged surface come from? This is this cuvette in which you have dispersed your sample, right? So what is electrophoresis? We need to remember it is the movement of the charged particle relative to the liquid it is suspended in. This is nothing but the electrophoretic mobility. And what is electroosmosis? It is the movement of the liquid itself relative to the stationary charged surface under the influence of an electric field. And this is called as electroosmotic mobility. So electroosmotic mobility needs to be removed from the electrokinetic effects so that we can measure the electrophoretic mobility accurately. So what happens is when an electric field is applied across the electrolyte, charged particles suspended in the electrolyte are attracted towards the electrodes of opposite charges. So if you apply positive volt to one of the electrodes, the negatively charged particles will get attracted, whereas the positively charged particles will be moved away from them. The particles move with a characteristic velocity, which is dependent upon the amount of voltage that you apply, the dielectric constant of the medium, the viscosity of the medium, and it depends upon the zeta potential. So the velocity of the particle in a unit electric field is referred to as electrophoretic mobility. What is electroosmosis? The walls of the capillary cell carry a surface charge. On application of the electric field needed to observe electrophoresis causes the liquid adjacent to the walls to undergo electroosmotic flow. So colloidal particles will be subject to this flow superimposed on the electrophoretic mobility. So apart from the movement of the particles, which is happening only because of electrophoretic mobility, electroosmosis also plays a role. So we have to avoid electroosmosis from taking place. So what we are saying here is you have an electrode. So this rectangular cuvette that you see here is again your electrode, is again your cuvette. you apply positive, you have two electrodes, as I said, positive and negative. This is negatively charged. This capillary tube that you have is basically negatively charged. So positive charges would be very close to the cell wall. So close to the cell wall, you will be having the electroosmotic effects, wherein the liquid will be moving with respect to the surface charges. But there will be a certain layer which is called as the stationary layer, in which this electroosmotic effects will not be taking place. And we have to determine this stationary layer in the older generation equipments so that we measure the electrophoretic mobility. So what we are trying to say is the electroosmosis effect takes place close to the cell wall. There is a certain layer in which the electroosmotic effects will not be taking place. So in the previous generation equipments, we had to find the stationary layer and do the measurements of zeta potential in the stationary layer, right? Now with the advancements in the technology, what we have done is we have come up with a technology mode called as the mixed mode measurement, with the help of which you can measure zeta potential at any point in the cell, right? You don't have to find the stationary layer, or the electroosmosis is compensated because of the mixed mode of measurement that we have. So what we are saying here is there's a point in the cell at which the electroosmotic flow is 0, and the measured particle velocity is the true electrophoretic velocity, which is called as the stationary layer. So in the previous graph, we'll see, so this is the stationary layer that you find, wherein the electroosmotic effects are 0, right? So this is the layer in which you will have true electrophoretic mobility. So in the previous generation equipments, you had to find the stationary layer and then do the measurements, then only you would get accurate measurements, so that you compensate for electroosmosis. So what we are doing is the measurement of particle electrophoresis would be made much simpler if electroosmosis could be avoided altogether. So the mobility of the particles at any point in the cell would then be the true mobility. This is what is done in the advanced DLS instruments, which use mixed mode of measurement using M3 pulse. So what we basically do is we do a fast field reversal. So if you look at this fast field reversal, as the frequency increases, your zeta potential value remains the same. So when you are changing the polarity of your zeta potential at a very high frequency, you are able to measure the zeta potential accurately, right? It remains constant. When you are measuring at low frequencies, your value of zeta potential decreases. So this is exactly similar to something that was happening during the multiple scattering in particle size measurement. So when you're doing at measurements at high frequencies, your value for zeta potential remains stable, but at lower frequency shifts, your value for zeta potential changes. So that is why in our instruments, we provide you with a fast field reversal and a slow field reversal. Why do we provide a fast field reversal? Because when you are doing fast field reversals at higher frequencies, the electroosmotic effects are not taking place. So you are able to measure zeta potential accurately at this place. But the resolution that we obtain at high frequencies is not very good. So the resolutions that we obtain at low frequencies are better than at high frequencies. So the measurement mode which we have is the mixed mode measurement, where we measure fast field as well as slow field reversal. The fast field measurement is done basically to avoid electroosmosis from taking place, and the slow field reversal is basically done so that we are able to measure the resolution of zeta potential in a better way. Okay. So fast field reversal in an alternating electric field is applied with sufficiently high frequency. The liquid velocity due to electroosmosis becomes insignificant with the respect to the electrophoretic mobility. So the measurements do not have to be taken in the stationary layer as in the previous generation equipments, and can be measured anywhere in the capillary cell. And as the FFR doesn't provide very good resolution, so we also do a SFR, which is a slow field reversal, so that we are able to do the measurements close to the cell wall for obtaining better resolution. So the mixed mode gives you a fast field reversal measurement to provide accuracy and stability of the result, and a slow field reversal measurement to improve the resolution of the methods that we have done. So this is how a cuvette looks like in our case. So as I said, here if you look at, there are two electrodes which are there across which you will apply the voltages. So you will apply positive voltage here and negative voltage here, and then you will reverse it. So reversing frequency is what defines the fast field reversal and the slow field reversal. So the fast field reversal will be basically done for measurement of zeta potential accurately without electroosmosis taking place, and slow field reversal will be done to measure the zeta distribution, so that you're able to measure with a better resolution the zeta potential values. So this is how zeta potential is done, and what are the considerations that you have to take care while you are doing zeta potential measurements. So this is it from my side. If anyone has any questions, I am open to the questions, both from particle size as well as zeta potential range. Any questions that are there, you can probably ask right now.
Thank you, sir. Thank you. It was a very elaborated and informative session. You have discussed about dynamic light scattering, history of DLS, considerations for the particle size selection, working of DLS, the concentration limits as well as particle limits for conducting DLS. You have nicely explained and highlighted various multiple scattering NIBS, that is non-invasive backscattering, its advantages, zeta potentials, dispersion stability, its mechanism as well as maintenance. You have also discussed electrophoresis and electroosmosis very nicely, as well as the fast field reversal. Now the session is open for the queries. Anyone having any kind of query, he or she can post their query in the chat box. We will unmute you. Anyone, either you can raise your hand also. I think, sir, there is no query.
Okay, ma'am.
Everything was very nicely and cleanly clear. Now it's time to felicitate Mr. Amit Bhatia. Sir, kindly accept our token of gratitude.
Thank you, ma'am. Thanks a lot.
Thank you, sir. Thanks a lot for sparing your valuable time with us. Now I invite Dr. Manpreet Kaur, Assistant Professor in Department of Pharmacognosy, for the vote of thanks.
Thanks, Arvinder. Am I audible?
Yes, ma'am, you are audible.
Good afternoon, everyone. It's my pleasure and privilege to present vote of thanks today. First of all, I'm very grateful to Dr. Indupal Kaur, Professor and Chairperson, UIPS, Punjab University, Chandigarh, for such a nice formal address. I wish to express my sincere thanks to Dr. Harinder Singh for delivering a knowledgeable talk on nutraceuticals, a suitable choice for pharmaceuticals. Further, I'd like to express my gratitude for Dr. Keza Raza for enlightening us on topic pharmacokinetics, an indispensable pharmaceutical tool. I would like to extend a special thanks to Dr. Amit Bhatia for giving insightful talk on basics and advancements in field of dynamic light scattering. I also wish to express special thanks to Dr. B.K. Kapoor for joining us today. At last, I, Dr. Manpreet Kaur, on the behalf of management, Honorable Principal Dr. Satwinder Kaur, faculty and participants, present heartfelt thanks to all eminent speakers for delivering such informative sessions and sharing with us important findings and opinions. Thanks, all of you. Have a good day. Over to you, Arvinder.
Thank you, ma'am. Thanks a lot. Assembly of FDP, we will meet. Thank you all.